Welcome to stream number 2.
Number 2 of an infinite number in the
Ooh,
future.
don't make promises.
Yeah. Today we're going to, we've picked a different citizen science project to last time, and we are going to be picking out plankton and classifying them on the Zooniverse.
Hey, before we get started, what's your name?
Oh no! My name's Trisha.
Nice to meet you, Trisha. I'm Michelle.
Nice to meet you too, Michelle. So yeah, we're 2 of the Science Scribbler team. We have our own projects on the Zooniverse, our citizen science projects, but that will come in the future. Today we're just going to be looking at one that someone else has made.
Yep. Yeah, let's jump right into it. So perfect. Trisha's gonna share the screen, and she's gonna start today. We've chosen Plankton Portal already because it looked really interesting. The plankton look really cool in the images. Yeah. But we know nothing about this, so we're also going to learn as we go.
Exactly. So no plankton equals no life in the ocean. Yeah, that's fair. Okay. So there's one workflow, and we've already gotten rid of the tutorial, so we're going to have to go through it. In this project, you'll be marking images of plankton, tiny oceanic organisms taken by an underwater imaging system. Okay, plankton, important food source. Yep.
Yeah. I want to know more about this underwater imaging system.
Yeah.
They really just skip over that part.
Oh, but they have really nice GIFs of how to do the task.
Yeah. Look at that. Okay.
Yeah, store that for later.
Yeah.
Yeah, select the shape that most closely resembles the plankton. Okay, I've learned something new already. Plankton being the single plankton.
Is that Latin or Greek?
I don't know, but is it in SpongeBob where there's a Mr. Plankton?
Yeah.
Yeah.
Oh, so he should be Mr. Plankton.
Perhaps. Yeah. Okay.
We'll write in.
Yeah.
This is the first campaign for our community.
Okay, select the shape in your image from the task menu, and then click on the plankton in your image and select the appropriate species. So we have a following task as well. Mark and identify all of the plankton in the image, then click finish. Others are classifying too. Yes, this is a great tutorial, animated tutorial.
Yeah, another GIF is great.
Yeah, okay, so we've got an example here of something which is a plankton and something which looks like background noise that isn't, that we shouldn't click. The wisdom of the crowd does give the right answer. Yep, wholeheartedly agree. Oh, and that is everything. Okay, fantastic.
All right. Simple.
More help, let's go. Okay, so
let's just go through the options on the right hand side there first.
Yeah.
So round with no tentacles, head with tail, jellyfish-like, ribbon/elongated, and bug-like. So let's check out the field guide. Those cartoons are nice, but let's have a quick look at some of the examples. So round, no tentacles. Okay, so really quite globular.
Yeah, so you've got lobate. My pronunciation of this is going to be wrong. So are they lobates or is it Latin?
Let's go with lobates. Yeah.
Lobates, round to oval with combs along the major axis. Okay. Larvation house, a striped blob-like web.
Okay, so kind of like a...
surrounding a tadpole-like inhabitant. Wow. Some images have been scaled down. Okay. All right.
Fine.
Let's have a look at head with tail.
Rocket ship.
I can actually see that this first one does look like a rocket ship. Yeah, it's a bit Star Trek, isn't it?
Yeah.
Thimble or triangle-shaped body, a long branching tail. Sidipid. Acorn or American football shaped with bands along the major axis.
Love that they specified American.
Yep. Sipho, corn on the cob. I can see that. I can see this one.
On the right-hand side one. Yeah. On a lot of the other ones, though, I don't. But
no, but this one is cob-like or like carrot-like maybe.
Yeah. Huh.
Diverse group. Okay, so those are going to look very different.
Yeah.
Sipho, two cups. Hooded organism. Okay, so
hold on. Go back up a little bit.
Alrighty.
I want to compare.
Yeah.
Oh, okay. So the top bit.
Yeah. So this little, I'm not going to say jellyfish-like because I think jellyfish is a category, but this kind of little umbrella that you see on top.
Yeah. Okay. Fine.
Yes, and then
Doliolid?
Yep.
Doliolid. That's a fun one. I like that.
Yeah. With tail. Okay. Barrel-shaped body, interior striations along the minor axis, and a pineapple in certain orientations. I can see that. I can see that here.
Yeah.
Oh, and this one especially.
Yeah.
Okay, let's have a look at the next. Jellyfish-like.
Yep, okay.
There we go. Okay, I see that.
Okay, so like really long tentacles, which is really useful to distinguish for the current one that's on screen, because I was worried that those little tiny bits that are running horizontal are tentacles, but I don't think so.
I think maybe those are combs.
Yeah,
from the first one.
From the first one, yeah. But
okay, Solmaris have a flat head, many distinct tentacles. Medusa have 4 tentacles.
Oh, exactly 4. Love it.
And then
more then.
Okay.
Fine.
Yeah. Medusa more than 4 tentacles, and Medusa goblet. Distinct 2 short, simple, symmetrical tentacles. They're typically shaped like a goblet. And then Medusa Ephirae.
Ephirae? That looks amazing.
Yeah.
Such a cool one.
How would you describe that? Like flowery almost? Or
there's like a, you know how you get those toys for like little kids that have like suckers? Yeah, exactly. That go onto like the windows or something. I'm pretty sure I have one that looks exactly like this.
Well, if we revisit this project, you'll have to bring that.
In.
Yeah. Okay, so we've done jellyfish-like, ribbon elongated.
Yeah. Oh, okay. Ooh, really dense.
Yeah.
Baroida.
Opaque, one rounded end. Hair-like appendages. Cestida. Ribbon-like, transparent. Okay, yeah, I can see that.
Radiolarian colonies. Extremely
variable. That's good news. Appears as a string or mass of circular bodies.
Okay.
And larvation, tadpole-like. That makes sense.
Arrowworm.
Okay.
And then fish larva. That's funny that it's like literally looks like a fish.
I think perhaps it is. I think in this instance, we're going to be. I would suspect that then this is a way of cleaning the data.
Yeah,
right?
No, exactly.
Because they'll have picked fish larvae as well.
Yeah. Yeah, well, exactly.
Yeah.
Yeah.
So only some of these are what they're looking for.
Exactly.
I would guess. Okay.
It's just fun that they've put it into the ribbon/elongated category.
And now we've got...
and then shrimp.
Yeah. Okay. So I know what a shrimp looks like.
Yeah.
Yeah.
Should be good.
Relatively confident on that.
Yeah.
Polychaete. Yes.
Worm.
Yeah.
Okay. Copepod.
Not copepod.
Yeah, copepod.
Yeah. Coping. An ovate body, antennae. Yeah. Very small. Good to know.
Ooh, this is the pterodactyl of the sea.
The pteropod. Very opaque, wing-like extensions and triangular-shaped shell. Okay.
Yeah.
All right. So why is a pterodactyl called a pterodactyl?
Oh, I have no idea.
Did it have a triangular head?
Oh, maybe. You think that is
I don't know. Or maybe it means wing. Wing-like extensions.
Yeah.
Or is anyway.
We might have to Google.
Yeah. Okay, so slightly more confident in what we're looking at now.
Okay.
So
this beautiful
example,
picture.
Yeah.
Right, yeah.
I would go round, no tentacles.
I agree. Oh, okay.
Oh, perfect.
Yeah,
okay.
And then we have to choose which one we think it is. I'm thinking Thalassophobia.
Yeah, I think so too.
The Ted Lasso.
Thalassophobia is the fear of deep water.
Isn't it? I have no idea.
I think. Yeah. Anyway, let's go with that one. Step is required. Okay, perfect.
Yeah. And
then I think there's nothing else there that I can see.
No. Not at the size.
So I'm going to go done.
Yeah. Ooh, okay.
Okay.
Do they have like a... There's nothing here?
There's unidentifiable, but I think if there's nothing here, we just go done.
Done.
Yeah.
Okay, yeah.
I don't think that the field guide has information on what to do with...
When there's nothing.
Yeah, these little bits. So I'm going to go through...
Oh, we got lucky with that first one. It had such a beautiful image on it. And I'm now concerned that we won't see any more.
Well, I think there's something here, but I don't
No,
think that
that's
we
too
can.
small. I think that's the background gunk.
Aha.
There we go. Ooh, I see tentacles.
I think we're perhaps going jellyfish-like for these ones.
I agree.
So, Medusa, more than 4 tentacles.
Is it?
Or Sulmaris.
Sulmaris.
It's
or
not
Medusa?
got a flat top.
Yeah, you're right.
So
okay.
yeah, if we're going, no, we're going to the jellyfish-like.
Yeah.
Flat
head, many distinct tentacles.
Or
versus
typical jellyfish body.
Yeah.
I think it's a typical jellyfish body.
I agree. Yeah. So let's do Medusa more than 4. And I think the same for that one. Yeah.
Yeah. Okay.
Gotta hit OK at the bottom. That's a bit of an annoying step because it's off the screen, but beautiful. Oh, they didn't really talk about this.
About what to do if the image is
if the
like thing
off
is at the end.
Edge. Yeah, exactly.
Well, I guess the good thing is that lots of people are looking at this, so we can make a
but
choice.
We can't, right? Because we don't know if it's flat or rounded.
Okay, that's true. In which case, I guess I would select unidentifiable.
Yeah, no, that's a good shout. Yep. So let's put it right at the edge there. Yeah.
Okay. I think there's nothing there.
Yeah.
Yeah.
Wonder what causes these streaks. It must be the camera, like a gain reference or something.
Yeah, we didn't learn about the camera, but I guess we can check that in a minute.
Oh yeah, why don't I'll go to on my phone, I'll go to the about section and maybe have a quick look. So before we learn about the camera, they're at 50% complete on this data set at the moment. There are just under 2,000 subjects, so images in Zooniverse terms. And they've had 6,000 volunteers so far help with this, which is great, for about 30,000 classifications. So we can maybe, it's a bit slow to update sometimes, so I don't know if we'll see our 5 classifications so far show up. But it is always fun to see the bar go. So let's go to the About page and just see if they have any information about the camera, because I think that's...
What do you think about this one? I think it might be Arrowworm, because it looks very similar to...
You know, I wouldn't have even, because that's so much smaller than the other one, that I wouldn't have even thought about that. Yeah, let's zoom in and see if it actually has any features.
Paired symmetrical fins. I actually think that these could be the fins.
You're right.
And it does look worm-like.
Yeah.
So I'm going to pick Arrowworm for this. So that was Ribbon. And then Arrowworm.
Yeah, good shout. I definitely didn't expect that to be so much smaller than some of the other ones that we had seen.
Yeah.
So I'm glad you noticed it. Okay, camera. Plankton is Greek. They answered our question.
There we go.
Yeah. Planktos, which means wanderer or drifter. Plankton vary widely in size from thousandths of an inch or less to several feet in length.
Really?
Greater than 1 meter.
Where do the ones that are really long live? I'm guessing further away from
It says
beach.
Most are less than an inch. Are you concerned? Do you have a beach holiday coming up?
I just don't, seaweed and all of those kinds of things don't really.
Yeah.
I don't think that's anything.
There are 2 main types of plankton: phytoplankton, which are plants, and zooplankton, which are animals. They are most interested in zooplankton. Their underwater camera system is optimized to capture images of larger planktonic organisms, ranging from a few millimeters to a few centimeters.
So visible, I guess it's an optical camera.
Yeah, it must be. Okay, how do we get the images? Here we
go. Perfect.
We got there.
Ask the imaging,
down a bunch.
The imaging scientist. How do you get the images?
How do the images happen?
Yes.
Okay, so this is jargon, and I think I might say it wrong, but it is called the in situ ichthyoplankton imaging system, ISIIS.
Okay.
Okay. Okay, so it's in some ways a virtual net with quotes around it, which captures the images of the organisms and information about their immediate surroundings. It samples continuously. So you end up with a collection of digital images that record the exact location of the various plankton organisms in relation to each other and the environment in which they live. The images are recorded onto a hard drive instead of their normal approach, which is to get a slurry of plankton all mixed together in a sample jar, and then they end up using formaldehyde to be able to like fix them and image them. Okay, so macro camera system, and it has its own illumination, and it's integrated into an underwater vehicle that has additional sensors for measuring depth, salinity, and temperature of the water, as well as dissolved oxygen, light level, and how much chlorophyll is there. Okay, so I feel like we missed a trick, and we probably should have done marine biology research. Because, like, how much fun would it be to, like, I mean, I'm sure it's hard work, right? But, out on a boat, if the weather is terrible, it must be, like, much, much worse. But
I think the field work is interesting, but if you're fixing plankton, you're probably counting them.
Yeah.
And what I know from environmental research and people who count pollen in a microscope, it takes a really long time and it's a very labor-intensive task, which is perhaps why we're doing it now with citizen science.
I mean, that's a good shout. I remember, because I worked with bacteria in my PhD, and there's a lot of counting bacterial colonies that goes into that. So okay, so yeah, it sounds like it's an optical camera that's part of the boat system itself, and it has its own lighting, and it's able to take images kind of, it says continuously, but it doesn't really talk about how that works exactly. But a very interesting system. Could maybe do with a gain reference of some kind, given the vertical streaks that we're seeing consistently in all of these images.
Yeah, do you think that that's just the background of the camera itself?
Yeah, exactly.
Yeah,
because it's showing up on every single image, which means that it's the exact same. It's not like real, essentially. So
you picked a good one.
Yeah, this is a fun one. I like it. They're just so pretty to look at as well. Like the shapes that they take, like it's like a little bit foreign, but like just like quite nice.
I think that might be something dead.
Like a fish? It has like a weird tail.
Yeah.
You think it's fish larva?
It could be bug-like. But I don't think it's big enough for us to be able to identify it.
Do you think it's that one with the tail? So the shrimp, like the from the right, second from the right.
Okay.
Yeah, or can we check out the fish one, fish larva?
That was in ribbon elongated.
Yeah, I think so. Just because that bit at the bottom, the back, looks like a little bit of a tail.
Yeah. I would go towards shrimp more than fish.
Yeah, no, I agree. Now that I've seen those images,
so let's pick
bug-like, yeah,
and go shrimp.
You're very good at spotting these small ones. Like even knowing the size variation that's possible, having read that introduction now, it always surprises me when you zoom in slightly and it turns into an actual shape. So, yeah.
I think we've got something here.
Ooh, yeah, I agree.
I'm gonna go worm.
Yep. Ribbon elongated.
And
colonies
arrowworm,
inside?
I think.
Yeah, arrowworm.
Yeah, no colonies inside.
Yeah, that's a fair shout.
I find it interesting that it does that here rather than
over on
going
the side.
through. Yeah.
Yeah, it is, like, rather than having it be a two-step process.
Yeah. So was there anything else? That looks like possibly it could be plant matter, just a guess.
I just saw on the ribbon elongated section there's a fish larvae option, which I don't think, was that in the field guide?
Yes.
Oh yes, it was. Okay.
Yes.
Yeah.
But I think, well, let's have a closer look and see if we can see the fins on this one, or if actually it could be fish larvae. I think I'm gonna go with ribbon elongated.
Yeah, no, I agree. I agree. You're right.
And I don't think there's anything else interesting here.
No.
Interesting, the field guide shows them all like this as well, which you often can see something different if you invert the image, or
that's true.
patterns will appear.
Yeah, I hadn't considered that.
Maybe you weren't there before.
Uh-oh. Okay. And then, so your marking has gone away.
But it's the same image, so let's try again.
Try again, yeah.
What's going on here?
Ooh,
oh dear. Okay.
That's it broke the GIF or the image.
Let's try something new.
Okay, hold on. Okay. So
it's kept that's really interesting. Yeah. Let's get rid of that mark. Then delete it there.
Yep.
And I think we're going to see a lot of the jellyfish-like medusas here.
Yeah, they do seem to be the most common.
With more than 4 tentacles.
Yeah.
And so in the about pages it said that they were interested in the zoo, the animal plankton.
Yeah, exactly. Yeah. And again, they didn't go into the details of like how, but they said that this imaging modality was like good at imaging specifically the zooplankton.
Yeah.
So not sure how.
Yeah, maybe to do with the detection of what it what it
or like movement with. Or something like that.
Yeah, or perhaps we're looking at stuff that's denser if we're talking about transmission imaging, so the light that's coming through the plankton, and so maybe the density of the plankton is important. I'm just thinking about X-rays.
I mean, I doubt this is X-ray imaging though.
No.
Yeah. Gonna guess optical.
It's optical, but still there's going to be a certain portion of light blocked depending on the material.
Yes. Yeah, that's true.
So this is confusing.
It's much larger than we've seen in some of the other ones.
I think that it still has tentacles. I don't think I would count these as combs.
No.
So
do you think it's flat or rounded?
I think perhaps it's still rounded.
Yeah.
But just really squished.
Yes.
Yeah.
Jellyfish, yeah. Oh, that's not what we... I was like, wait a second. There we go. Empty image.
Yeah. Oh. The importance of scrolling up.
Yeah. Yeah, it is like just slightly the wrong size.
Okay.
Yeah.
We're getting a lot of jellyfish like, which is okay.
Yeah. I love how we saw the first jellyfish like and we were like, ooh, so pretty, so pretty. And now we're like, yeah, we just want something else.
Yeah, because
give us something new.
We can look at our stats and how many we've classified today, but I actually think we have something new here. I think that's combs. So round, no tentacles.
Yeah.
And then what is the question then?
The lasso, I think, yeah.
It looks closest to that, yeah.
There's kind of that middle bit, though. It's like a mushroom.
Comb rows along the major axis. Balloon shaped. We don't have that. There's no... Is there a tadpole-like inhabitant here? I don't think that would count.
Mm-mm.
Can be very transparent. Oh, interesting.
That's it. Those are the options. Can you invert it just so it looks more similar to the images in the field guide? Let's go to the jellyfish.
Yeah. These are, I guess the tentacles vary from short to very long.
Yeah, it's got like that internal structure inside of it though. That's the part that's...
Ooh. Oh no.
Uh-oh.
This that I would call a lobe, if I were to have to guess. Found a tricky one.
Mm-hmm.
What do we think about spending too long on classifications?
Yeah, that's always a little bit tricky because I think the longer you spend on things, the less sure you are. Which I don't know if they use that information, but that could be interesting.
Yeah.
They probably also just see it in the data itself because there'll be more confusion, so people will choose many different things, and then it'll be clear that there's less consensus.
Yeah.
So.
What's your gut saying?
My gut is saying jellyfish, but
okay. Yeah.
I don't know, because it doesn't really have the tentacles. Like maybe there's... Can you zoom in? Because there's like one longer thing.
Ooh. I can zoom in.
You are capable. You are strong.
I can do this.
Yeah.
It almost looks like a fish with a jellyfish on the outside, doesn't it?
Yeah.
With this central bit.
There's definitely something inside of it, which is where like that's part is what's tripping me up because it doesn't look like a comb.
Let's look at head with tail.
Okay.
Process of elimination. None of those.
None of those.
None of those.
Yeah.
Okay. Ribbon elongated. Don't think it's going to be one of those.
No.
Pretty confident.
Yeah.
Bug-like. Nope.
No.
Okay. So
ribbon, no tentacles, or jellyfish-like. Let's look at the combs again on the ribbon, the round, no tentacles.
Bristled rows along the major axis. This is the thing that...
But none of those particularly show the bristle,
no.
So it doesn't feel like it's that. I think it's a jellyfish. I think it's just a jellyfish with really short tentacles.
A medusa with more than 4 tentacles.
I think so, yeah.
Yeah, okay. Yeah. I really like the sketches they've got as well for this.
Yeah.
It's all visually very nice.
Mm-hmm. So when you zoomed in, like, did we see any? I thought I saw another. Yeah, that thing.
Well,
little tiny one.
I don't think that's the only thing to look at, isn't it?
Yeah, I think so.
I can't identify any features on this.
Oh yeah, no. Now that we've zoomed in, I agree. That's a little too blobby, so. Oh, it stays inverted. That's nice. I thought you were gonna have to click it for every single one, so.
So we've got something different here, I think, up in the corner.
Oh yeah.
But I don't think we're going to be able to identify it.
No, we don't have enough information.
So interesting, but not
exciting.
No, I didn't want to say it wasn't exciting, but
I'll say the
intriguing but not satisfying.
There you go.
I think all of these things are too small.
Yeah.
I don't think anything's there. And again, which way do you prefer it?
So I was actually thinking this to myself because the uninverted looks most similar to the imaging that I am used to from like the cryo-EM, cryo-ET like type of imaging.
This one?
Yeah, this one, where it's like grayscale and the things that you are interested in show up dark. And I just want to point out that that's like a transmission thing. So like when you actually are taking the image, the electrons have to pass through like the feature of interest, the sample, and then you collect them on the other side at the detector. And so where something is present and dense is where the electrons don't make it through, and so that shows up as dark.
So you're looking at a shadow.
You're looking at a shadow, essentially. Yeah, exactly. And so that's like a very traditional electron microscopy viewing experience. You can always invert your images, and some people used to do that for data processing for historical reasons. But like the actual images themselves, when you are looking at them, like this is the style that I'm most used to because of that. I doubt that that's like the case necessarily for this optical imaging in terms of transmission and all of that. We don't have enough information to actually say it. But this is the style that I'm most used to. But they show all of their images and their entire field guide and all of that using inverted. And so it's almost easier sometimes to compare with the inverted.
Is it because, do you think that it's shown inverted because we assume that the sea is dark underwater?
Do we assume that?
Do we? That's a good question.
do you assume that?
I've never been deep down into the sea.
I mean, I know it's dark, like once you go very, very far down. Like, I think that's common knowledge, but I wouldn't have necessarily made that connection here.
This is more similar to the imaging that I'm used to, though, because I did a lot of CT scans, X-ray CT scans of porous sponges, essentially. Yeah. And so almost 98-99% of the X-rays get through, and so you're looking for that one speck of white. Yeah. So I'm used to analyzing things that are like white. This. Okay. Yeah.
Interesting. Yeah, that is interesting. That would be like a fun like icebreaker for a microscopy conference.
Well, maybe it should be which of these two images do you prefer, inverted or not? And do you have your computer in dark mode at all times? Like what color is the background of your coding? Yeah. Is it white or black? Yeah. We've got another one of these. So this looks very similar to the one we ummed and ahed over, but actually
it's got longer tentacles.
Yeah, and looking at this one, which I think is jellyfish-like, you can see that inner lobe.
Absolutely.
So I think I'm confident now that we've made the correct assessment.
3 images on.
Yeah. Well, let's talk about learning then.
Yeah.
And can you learn as you go, and can you improve, and how do you improve when you're doing these projects?
Yeah. I mean, I think that you can definitely learn as you go. The more exposure you have, the more likely you are to like see things and like be able to interpret them in new ways. Like, for example, I find it really helpful when you're first starting as well to like do this with someone else, because like you saw the really tiny like one early on, like the fish larva or whatever it was, and like I would have 100% just clicked past that, like, and not even realized it was a thing that we should care about. So like, you know, you can learn, but it also sometimes takes an additional perspective.
Yeah.
And so that's where it can be helpful sometimes on the talk boards, because you can, so we've just been clicking done, but you can also click done and talk, and then that tags that specific image. It puts it onto the talk boards, and you can like write a little message or like do a hashtag tag or something like that.
Yeah.
And like make it clear for other people sometimes. And then if they spend some time on the talk boards, they might see things that either they missed or that others are seeing that then will help them in the future as well. So
let's take this one for example then. I think this one is something that looks pretty confusing. We've had one that looks a little bit like this before.
Yeah.
I think we're going to end up with jellyfish like, which is seems to be the popular choice class of plankton at the moment. But
I think the big question though is if it's that flat or if it's rounded.
Yeah.
So Solmaris or Medusa.
Let's have a look then
at the field guide. Yeah.
Solmaris looks so distinctly flat in the ones that they've given, I guess with the exception of this one here.
Yeah. It does seem to have more like kind of ring structures that are at that top area as well. Like it looks kind of like a lamp almost, like a lamp shade.
Yeah.
With like
tasseled lampshade. Yeah.
That you'll find in your grandma's or something. So yeah, I think this one's probably rounded rather than Solmaris.
And it looks like often occurs with multiple as well. So maybe that's giving an indication that this isn't it.
No.
Catch-all for jellyfish that don't fit into 1 or 3. Okay, so what is Medusa 1 and 3? Okay. Anyway.
They've used a terminology there that we don't
clear.
know. Yeah. Okay.
I wonder if this is one where we can have a look and see whether or not other people have been confused by this image. So sometimes the website will have that talkboard built in now into this classification interface, so you can have a look in real time at what other people have said about this.
Yeah.
But it's not fully
embedded.
It's not embedded everywhere. Yeah.
Well, let's finish this one up and maybe go have a look at the talkboards.
Alrighty. Has anyone said anything?
About
No.
This? No. Add a note about this subject. Okay. That's fine. Well, we don't necessarily have anything that we want to say here, but let's have a look at talk in general. So notes, comments about individual subjects, and then there's some general science and some troubleshooting. So let's have a look at the notes, I think. There are 2 arrow worms and a few copepods. Ooh, let's look at that one, the top one there.
Oh, this is a very thorough contributor, volunteer.
Oh, and those are tiny as well. So one of the features that maybe is slightly missing here is that we can't actually see what this person has annotated for this image. So they have to actually describe it using words in their post. So they've said notable features: lower left quadrant contains 2 distinguishable shapes. One curved, potentially a larval organism or a detritus, and one elongated linear form, possibly marine snow or small zooplankton. No large or clearly identifiable planktonic organisms. And then they use some very specific words for that. And then someone else disagrees, and they said there are 2 #arrowworm and few #copepod. So I thought I saw an arrowworm as well when I first glanced at it. I wasn't quite sure about a copepod though. I don't know what that is exactly. We might have a look at the field guide again.
Yeah, one thing we can't do in this is zoom in. So I think that here we've got the arrowworm.
Oh, I was going to say the arrowworm was the very straight thing.
So then 2.
So let's have a look at the field guide real quick, because now I'm curious. So what is, let's go to...
Do you want to look up a copepod?
Ribbon/elongated. So yeah, arrowworm.
So if we look at this bit here with the fins, the
you're right. Yeah,
I think that this is slightly out of frame.
Yeah, it's like kind of kicking its tail up. Yeah, but yes, I agree now. Okay.
So I see 2 arrowworms. Yeah. And what did the second?
Copepod, which is not a term in like the field guide. So let's do a quick Google.
There are 2 arrowworm. I think that it is in the field guide.
Is it?
In the final, it could be bug-like. Yes.
Oh, okay.
Wow.
You've got better memory than I do. Oh, and they think that the very straight.
So the second person said that there are 2 arrowworms, which I think we think are these.
Okay. And they also think
several
copepods.
Yeah.
These must be minuscule, like super tiny. How were they able to see this even in the image without being able to zoom in? Okay, now I'm worried that we missed things. I think we would have gotten these 2 arrowworms, but I'm not sure that I would have seen a copepod.
I think I wouldn't have looked at many of these in great detail. Mm-hmm. Yeah. Let's have a look again at the scale. So they're very small, and this circle here is looking at the size difference. Yeah. That's somewhere in the, I think it might be here.
They had to like zoom in for some, right?
Yeah.
Some images have been scaled down in order to provide a sense of scale. Each image has a size marker.
Okay, so that's a little bit counterintuitive to this. I'm going to have to think through exactly what this means. The ones which are a smaller circle are bigger. Is
a larger specimen, yeah. And they've actually made the image like smaller so that it doesn't take up as much space.
Okay, so then if we're looking at, can we do a direct comparison between what we're seeing here and what we're seeing here on these 100% ones?
Okay. Yeah, that's a really interesting approach. So the bug likes. Okay, so those that's like a full scale image on the right hand side.
Yeah.
2 of those. Okay, so that's the size that we would expect them to be. So yeah, we're looking at some of these like larger pieces of what I would have said detritus or snow or something like that for this scale. But,
marine gunk.
Yeah.
Yeah.
Okay. I mean, that's definitely really interesting to look at and just kind of get a feel for things that we might have missed. So, should we have a look at one more note before we finish up for the day?
Maybe we should look for a rocket ship tag.
And
see if we can find any
Absolutely.
subjects that have rocket ships in them.
Yeah. Oh, these look completely different. Yeah.
Okay. A siphonophore. Okay.
Oh, interesting. Okay.
So this is a bigger image.
It is, yeah. I believe you're talking about that round guy in the lower center with a short tail. It is a small siphonophore, the hashtag rocket ship SIFO type.
See the second image example for rocket ship.
Okay.
Where are we going into? Is it head with tail? Yes.
Yes, yeah. Good memory.
Second image example for
SIFO type.
Rocket ship SIFO type? So
yes,
is that this one here?
Right. Yeah, it must be.
Okay.
Okay, so like 2 or 3 little round. Yeah, okay.
I can see that.
Yeah, I can see that as well. Yeah. Okay. And then there's
kind of
there's a couple
like
of like little
like spindly things.
Groupings of them, yeah. I don't know what that is exactly.
Let's have a look. Nothing else mentioned, so maybe there is just the one in this image.
Yeah. I'd be surprised if there wasn't quite a lot of like little tentacles floating around.
Yeah. There's just stuff. Isn't the sea full of stuff? I wonder how much of this is plastic.
Ooh.
Yeah.
Yeah. Shall we leave on that upsetting note?
We can. We can leave on that upsetting note.
All right.
Right.
Well, this was lovely. We'll see you all again in a couple of weeks.
When we next schedule it. Yeah. Yes. We will schedule the stream so you'll be able to see on the countdown when we're next going to stream. Probably a Friday afternoon UK time. If you have any requests for Zooniverse projects that you would like us to do as part of this, leave them somewhere that we will see them. Yep. Or no, I guess any Zooniverse project that is a Science Scribbler project, you can comment a question for researchers. On Twitch? Can you comment on Twitch? I never have because I don't do that. I interact with the live chat, but yeah. All right. Maybe comment on a VOD. I'm not sure. Yeah. I will turn this into a VOD. So yeah. Yep. And then we can see what we can do. And if you have any suggestions for projects where you would like the researcher themselves to come on, we're starting to think about having the researchers come on to do Q&As with us as part of this and to explain a bit more than we're capable of from the about pages. Yep. So drop us a line and we'll see what we can do. All right. That's it. Yeah. Thanks all. Until next time. Bye. Bye.